Hello, everyone, and welcome to our MGUS Smoldering chapter. My name is Audrey Burton-Bethke, and I am the Myeloma Crowd Community Manager. The event today is brought to you by the Myeloma Crowd Community Program by HealthTree. I'm going to be sharing some slides with you today in order to just give some intro announcements, and then we can get started. Um, Okay, so today's session will be recorded, and the recording, along with my slides, will be sent to you within, and all registrants within 48 hours of the event's conclusion. A very special thank you to our sponsors, without whom this would not be possible: Bristol Myers Squibb, Amgen Oncology, Karyopharm Therapeutics, AbbVie Adaptive Biotechnologies, Genentech Oncopeptides, and Takeda Oncology. Today's topic is clinical trials for high-risk smoldering myeloma patients. Last session in this chapter was our launch, and we kicked it off by having a presentation from Dr. Irene Gabriel. She talked about her precursor myeloma research and why patients' contributions to critical myeloma research is so important into leading us closer to a cure. Today, we have invited Dr. Jeffrey Zonder to speak to us about clinical trials that are available for the smoldering myeloma patients, specifically those who are high risk. Clinical trials are one of the key ways that we can contribute to critical myeloma research and accelerate a cure. It is now my pleasure to introduce Dr. Jeffrey Zonder to you. He is a hematology specialist in Detroit, Michigan, and has over 26 years of experience in the medical field. He graduated from Wayne State University School of Medicine Medical School in 1995. His interest in multiple myeloma and plasma cell disorders and diseases dates back to medical school and remains the focus of his clinical practice and research presently. He has developed and led clinical trials investigating new therapies and novel combinations of medications for patients with all stages of myeloma, as well as patients with primary systematic amyloidosis. So, Dr. Zonder, at this time, if you want to turn on your camera, and I'll just start with that question that I had for you. How has your interest in high risk smoldering myeloma changed over the years, and why is it one of your interests?
Thanks. First, I'd like to thank you very much for the invitation to speak with everybody today. And I have to admit, I can't believe you're springing on me now that I'm following Irene Gabriel,
Sorry.
as the next lecturer. But thank you. She's a fantastic speaker, and this is her area of clinical focus, research focus. My interest in this space, I think, has evolved um, as our understanding of this space has evolved. The fact of the matter is that we have really, as I'm going to talk about this in some detail during my slides, but our understanding of how plasma cell diseases evolve from a precursor condition that has relatively low risk to a precursor condition with higher risk to actual myeloma
Mhm.
or related conditions. Um, we have teased apart a lot of the biology related to those changes or that progression, and it's been it's first of all the field the biology is fascinating, but I'm also fascinated by sort of the way the research community's attitude, but also patient attitudes have evolved over time.
Mhm.
Um, in terms of their interest in treating this, by definition, asymptomatic, condition.
Mhm.
And like I said, we'll talk all about that, during tonight's chat.
Great. Okay. If you want to pull up your slides then, and we can get started. And thank you to all who are joining us during this holiday weekend. We know that this kind of includes the holiday, and we're grateful for all of you who are here joining us today. Um, Yeah, I know it's been a crazy day for both Dr. Zonder and I, so we're happy to be here with you.
Can we see my
Yep.
slides?
Yep, they're perfectly visible.
All right. So thank you. My talk probably will be no more than half an hour in length, and then that will leave us plenty of time to chat. And I view that as important, that time to chat, because the more people who are listening to a lecture, the harder it is to actually meet everybody's needs. And in support group type settings, the range of knowledge and sort of where you're at in your journey is huge. And so there are some people who really need like smoldering myeloma, MGUS 101, and there are some patients who want very, very detailed information that most lay people don't really even have an understanding of. And it's hard to put all of that in a single talk. So my strategy is to design a talk that has general, oh, if it lets me advance my slides, general topics and objectives, and then maybe introduce some very specific things that we can talk about during questions and answers, but not to dive too deeply into them in case there's not general audience interest in those things. So the first thing I'm going to do is sort of define what MGUS, or monoclonal gammopathy of uncertain significance, is, how it's distinguished from smoldering myeloma, how both of those are different than treatment requiring multiple myeloma, talk about risk stratification within each of these groups and why it matters. We'll discuss the results of really mostly one key smoldering myeloma trial. And then I'm just going to mention some trials that are ongoing and then answer any questions that you have. So monoclonal gammopathy of uncertain significance, that's what MGUS stands for. Monoclonal gammopathy means that you have an abnormal protein in your blood, an abnormal antibody type protein that's made by an abnormal clone of plasma cells in your bone marrow. And it is it's again, by definition, it's an asymptomatic disease that is not causing any symptoms at all or any problems for you at that moment. But there's a subset, and that's sort of illustrated in this cartoon, a subset of patients who go on to develop multiple myeloma or related diseases. And it's not the majority of these patients, it's really a small fraction each year. We define MGUS or myeloma or a middle condition, smoldering myeloma. That's what SMM stands for here, smoldering multiple myeloma, in part on the number of plasma cells or abnormal plasma cells that are in the patient's bone marrow. When somebody has, so a normal number of plasma cells is less than 5%. And usually it's a mixture of different unrelated plasma cells, different clones of plasma cells. We call that multiclonal or polyclonal. But if one bad actor starts proliferating or overgrowing at a higher rate and becomes a sort of a dominant plasma cell, it gives rise to a dominant plasma cell population that's all derived from that one abnormal cell. We call that clonal or monoclonal. And if the number of plasma cells in the bone marrow increases to over 10% or more, that by definition is multiple myeloma. But we call it smoldering myeloma if there are no symptoms related to it. So the chances of having symptoms increases as you get further and further north of 10% plasma cells. And when I say 10%, what we do is we do a bone marrow biopsy and we're looking at the total number of cells in the bone marrow space. And of those cells that we count, what percentage of them are plasma cells? So when that percentage crosses 10, we call it myeloma. If there's no symptoms, we refer to that as smoldering myeloma, until we get all the way up to 60% plasma cells. And for reasons I'll explain in a minute, once we hit 60% plasma cells, even if there are no symptoms, which is actually rather unlikely, but even if there aren't symptoms, we consider that to be treatment requiring multiple myeloma. Usually, before people hit that 60% mark, they develop symptoms, and the mnemonic that we teach students and residents and fellows and patients is CRAB criteria. And CRAB is an abbreviation that stands for calcium elevation, renal injury, or that's kidney injury, anemia, or bone lesions. So that's CRAB. And if you develop any of those, then by definition, you have myeloma symptoms, unless you can explain the symptom due to some other underlying condition. But also, as I mentioned, there are other criteria like that 60% bone marrow plasma cell mark, an extremely elevated free light chain ratio. And free light chains are like fragments of antibodies, of M protein antibodies. And it's common that patients with MGUS or smoldering myeloma or myeloma have more than one blood marker. They have an M protein, and then they have this other marker called the free light chains ratio. And if that's extremely elevated, that can be criteria for treatment requiring myeloma. And then, of course, bone lesions identified by more sensitive tests beyond just x-rays. The original CRAB criteria defined the bone lesions as ones that you saw on standard x-rays. And of course, we've realized as our imaging modalities have improved, MRIs, CTs, PET CTs, we've realized that we can find bone lesions in patients that you don't see them with x-rays. And I'll show you a study in where this had relevance in a little bit. But these last three criteria actually were more recently added. At this point, it's really several years ago, but they were added because studies done showed that if you had any of these last three things, even if you didn't have CRAB criteria at that moment, you had about an 80% chance of developing CRAB criteria in the next 2 years. And it was felt that that was sufficiently high risk that it warranted starting treatment. So the definition of treatment requiring myeloma evolved based on that. The way I drew those last pictures, that diagram of what happens is that the progression from MGUS to smoldering myeloma to myeloma was purposeful, because we have to think of these as a spectrum of disease. All cases of smoldering myeloma or multiple myeloma develop from a pre-existing case of MGUS, but because MGUS doesn't have symptoms, sometimes we don't actually recognize that a person has MGUS until they've developed the symptomatic disease. We know about this association because we've had the opportunity to do studies in myeloma patients who, for one reason or another, had stored blood, and that blood was able to be analyzed to look for the presence of an M protein or an elevated light chain. So let's talk about MGUS first. MGUS is the earlier precursor condition. And when we look at that, there is a staging system, a low, medium, and high risk staging system. And the way that we stage MGUS, again, by definition, less than 10% bone marrow plasma cells, usually an M protein of less than 3.0 grams per deciliter. If the we have two basic diagnostic criteria that we look at. The M spike, is it greater than 1.5 grams? and the free light chain ratio. Again, it's the measurement of this free light chain particle fragment. Is it abnormal? If neither of these is the case, then the 20-year risk of progression to multiple myeloma is actually less than 0.5% per year. If you have either one of those two things, your risk of going on to develop myeloma is about 1% per year. And it's slightly higher than that, about 1.5% per year if you have both of those things. Okay. So again, as I said, a small subset of MGUS patients go on to develop symptomatic myeloma. Now, what about smoldering myeloma patients? These are patients who have that higher plasma cell burden. So we use something in the United States, we commonly use something called the 20-20 criteria. And this was derived at the Mayo Clinic. And what this mnemonic 20-20 is to remind us 20% or greater bone marrow plasma cells, 2 gram or higher M spike, and a free light chain ratio of over 20, 20 or higher. And what the model tells us is that if you have none of these three things, your 5-year risk of progression to symptomatic disease is about 23%. So it's between 4 and 5% per year. If you have one of them, you're considered intermediate risk, and just less than half of patients will progress to myeloma in 5 years. And if you actually have 2 or 3 of these features, 80% of patients, 81% of patients will progress to myeloma in the next 5 years. Okay, so your risk is quite different, 4 times as high if you have 2 or 3 of these factors than if you have none of them. And these could all, these are all patients who meet the current definition of smoldering myeloma, but these very easy to measure clinical factors put them into completely different subgroups in terms of risk. This is, just to sort of underscore that, and also just to point out the fact that even the lowest risk smoldering myeloma has 2 to 3 times the risk of progression that the highest risk MGUS does. So this intermediate group really is different biologically. These are patients who, as a group, are more on their way to developing symptoms, particularly patients with intermediate or high risk smoldering myeloma. So why do we care about this? Like what's the, why do researchers and why should patients care about their risk stratification, particularly in that smoldering myeloma space? The absolute difference in risk annually in low risk versus high risk MGUS is only a couple % per year, but the risk in low risk, the difference in low risk compared to high risk smoldering myeloma is about 10% difference per year or more. So I mean, it's a big difference. There's a huge difference in risk. So one reason we care is when we're comparing studies, right? So if you're looking at the results of 2 different clinical trials, and you're trying to figure out, does one seem more promising than the other? How do the results of trial A compared to trial B? Any statistician would tell you you can never do that. Okay, it's not statistically valid to compare the results of studies directly in a sort of statistical or mathematical way, but you can eyeball a study and quickly identify if there are major differences in the study populations, which could at least be contributing to differences in outcomes. So for instance, if you had study A, which was 90% patients with low risk smoldering myeloma, and study B, which was 90% patients with high risk myeloma, you would expect 3 times as many patients to progress in the second trial. And that doesn't actually mean that the therapy that you're using is any better or worse than the same therapy used in lower risk patients. There's just different biology in those groups. So you have to know what your baseline expectations are for the group you're treating when you start a study, and you have to know if you have similar baseline expectations when you're comparing the results of 2 studies. Next, it's important when we design studies. So the size of a trial depends on the magnitude of expected effect. So another way of saying this is when you're trying to prove a very small difference in benefit between one therapy and another, you actually need a very large number of patients to recognize that. If something works 10 times as well as the other, you can sometimes prove that with, 1 or 2 dozen patients. But if something works 1 or 2% better, you might need hundreds of patients, or even 1,000 patients to prove that there's a difference between the therapies. And so, knowing this, having awareness of what your baseline expectations are, and then being able to estimate the magnitude of benefit that you're trying to establish, that helps you design a study with the lowest likelihood of giving you an incorrect answer because it's not a powerful enough study to actually answer the question you want to ask. Very, very important. And many good studies, otherwise good studies, have been foiled by faulty statistical assumptions during the design phase. And also, finally, it helps us make clinical decisions because I'm going to show you the results of a study that basically demonstrated that the benefit seen with a particular treatment was really limited to one subset of patients and not to all patients across the board who were enrolled on a particular trial. And so if you want to use the results of that trial and apply them to clinical practice, you have to actually know which subgroup the patient you're talking to falls into. Okay, so treating high-risk smoldering myeloma. I'm going to show you the results of 2 randomized studies that have really helped establish the only current standard therapy for smoldering myeloma. The first was a Spanish study looking at 119 patients with smoldering myeloma, so a relatively small trial. And these were high risk according to either the Mayo Clinic's risk stratification system at the time this study was designed, or the Spanish myeloma group's definition of high risk smoldering myeloma, which was a little bit different. And they permitted patients who were considered high risk by either criteria, and they randomized them to either lenalidomide and dexamethasone. So lenalidomide, the other name for that's Revlimid. So that's a standard myeloma therapy, but it isn't approved for smoldering myeloma, followed by just lenalidomide. And that was compared to just observation, which was the standard of care for smoldering myeloma or any asymptomatic plasma cell disorder. And patients were followed until they had disease progression to treatment requiring multiple myeloma. And what this study basically showed was, at the time it was initially reported, the average time to progression in the observation group was 21 months. So a little bit less than 2 years, which is which tells you that this was really a pretty high risk group, right? Remember I told you that high risk myeloma by the 2220 criteria, it's about 16% per year. So this group, which again enrolled patients according to criteria other than just the Mayo one. They used the Spanish criteria also. This behaved in an even more aggressive fashion than would have been expected. But the median time to progression, that is 50% of patients at the time the study was reported, had not been achieved in the group that got therapy. And actually, there was also a slight survival benefit favoring the group that got lenalidomide, which was unexpected and actually was felt to reflect a bit of a flaw in this study that perhaps patients who showed evidence of progression, either by, particularly by labs, that would have led an average myeloma physician to start treatment. In this particular trial, they did not necessarily start treatment until they actually developed symptoms rather than just progression of their labs. And that was felt to perhaps contribute to the survival difference and may have reflected under treatment according to sort of American standards about when a patient would start treatment. Now, there was another study done, a United States counterpart done in the United States, which was slightly larger, 182 patients. And this was a randomization of lenalidomide given for 3 out of every 4 weeks, the way we typically use it in myeloma, although without steroids, versus observation. And again, the endpoint was progressive disease. And this study also showed benefit favoring the treatment, although it showed sort of, I think, progression in the observation arm that was more consistent with what we expect according to the 2220 criteria, with about 1/3 of patients progressing at the 3-year mark. So this is about a 10% per year progression rate, which is right about what you'd expect for intermediate risk and slightly below high risk myeloma according to the 2220 criteria. And this was inferior to what we saw with lenalidomide, where only 10% of patients, just less than 10% of patients progressed to needing treatment by the 3-year mark, even though the vast majority of patients never achieved a complete remission on this study. Therapy was finite, even though patients were permitted to be treated until progressive disease. For one reason or another, some patients on this study discontinued therapy before they actually showed progression because of things like side effects, and there was still this benefit noted. So when they did a subgroup analysis, it became clear that really all of the difference between these two arms was really limited to the patients who met the definition for high-risk smoldering myeloma. The study permitted intermediate or high-risk patients to be enrolled, but when you looked at just the intermediate risk patients, there really wasn't very much of a difference, at least not a statistically significant difference in the rate of progression, but there was a significant and statistically meaningful difference in the high-risk group. And so because of this, we generally view lenalidomide or lenalidomide and dexamethasone as therapy that is appropriate to consider for patients with high-risk smoldering myeloma according to the 2220 criteria, but generally not for patients with lower risk smoldering myeloma, with the possible exception of patients whose M protein is increasing over time, even though they don't meet the criteria for high-risk smoldering. So open questions at the moment, okay, just open general areas of query. Going back to this design, this diagram that I showed you before, the first question one might wonder is, who develops MGUS in the first place? And it's very typical and I think attractive to look at patient factors and environmental factors. So genetic inheritance and predisposition, and also environmental exposures. Are those how you end up in this larger gray circle to begin with? We don't, I'll talk about some examples of the kind of research that's being done with that. The next place you might ask a question about is, what causes this small subset to actually progress? And this area of research generally focuses on plasma cell genetics and molecular biology, but also patient immunity and immune surveillance, and whether there are reasons in the patient's bone marrow or immune system that allow MGUS cells to sort of escape and become malignant. So let's tackle the first one first. So MGUS, there's a very slight excess of cases in men compared to women. We know that the frequency increases according to age. Our first glimpse of this information came from a bank of specimens started decades ago at the Mayo Clinic in Olmsted County, where all residents of Olmsted County were basically screened for monoclonal gammopathy. And what they saw was the incidence, the background incidence in people who had no symptoms and weren't expected to have myeloma or any condition, it was about 3% in people who are 50 years of age or older. But by the time you got to patients who were only 85 years of age or older, that increased to about 7.5% of patients in that age group. So it becomes increasingly common as people get older. And as our testing for it has gotten more sensitive, one example is our ability to check for free light chains, something that I mentioned already. The incidence actually is even higher. Now, the story gets even more interesting when you start looking at ethnicity. And it turns out that people of African lineage have a higher incidence of MGUS. It's actually twice as high as the incidence in people of European descent. So one could ask, is that a genetic predisposition, or is that an environmental exposure? In countries where populations tend to still live in somewhat segregated groups, that might be hard to tease apart. However, if you look at the incidence of monoclonal gammopathies in people of African lineage, no matter where they live in the world, it does seem to be higher everywhere. And so if this fact is true in people who currently live in Africa, Western Africa, and true of people who live in South America and the Caribbean and the United States, it's not really plausible that that's an environmental exposure. So there does seem to be some genetic predisposition to this. Interestingly, there doesn't seem to be a particular difference in the rate at which MGUS or smoldering myeloma progresses to myeloma between people of African lineage and people of European lineage. So although there is a higher incidence of myeloma, just as there is a difference in the incidence of MGUS, it seems to be proportionate. The rate of developing a monoclonal disorder, monoclonal gammopathy or plasma cell disorder in the first place is what seems to be driving the increased incidence of myeloma later in the same patient population. So we and others have looked at this. So we teamed up with multiple other centers to do something called a genome-wide association study, which looks at genetic variation in the study that we did, genetic variation in African Americans and Caucasian people who did and didn't have multiple myeloma in each group. And we looked at variations of genes across the whole genome in each sample. And we tested samples from about 10,000 people and found some evidence of gene variations which may impart some risk or some predisposition for developing multiple myeloma. But none of them had extreme predictive power. It wasn't like, you inherit a certain gene and you will have a certain disease. So a great example of a one gene, one disease is sickle cell anemia, which is another disease that disproportionately affects African American people. If you have one copy of a particular gene, you have sickle cell trait, and if you inherit two copies of that, you have sickle cell anemia. There's no gene like that in multiple myeloma. At most, there are genes which somehow slightly alter risk. Another example of an important study looking at inherited risk is the PROMISE study. And this is probably something you heard about when Dr. Gobriel spoke with you. This is a screening study for patients, for high-risk population. Basically, patients or people who are between the age of 40 and 75 who are either African American or have a relative who is affected with MGUS myeloma, smoldering myeloma, or a related condition. You are eligible to potentially be screened and donate blood and be examined to see if you have any evidence of a monoclonal plasma cell disorder. And this trial will eventually screen 50,000 people, and the plan is to screen this many people to identify 3,000 people, and then the study will be looking at, among other things, what is different about the 3,000 who have these diseases versus the patients who don't, and then ultimately later, what is different about the patients who are identified as having one of these precursor conditions who go on to develop myeloma versus those who sit tight and just have an asymptomatic disease. Now, remember, the second part of the question, the second open question I asked is, what drives the progression from MGUS to symptomatic myeloma? And it depends. You have to think about progression. Our understanding of the sort of our working model of how cancer progresses has become a lot more sophisticated over the years. This particular slide just only scratches the surface of it, but I sort of have two diagrams or cartoons which I think illustrate kind of a key change in our understanding of this disease, and also has, I think, really changed our interest or driven our interest in earlier treatment. So the original model that we used to think about was sort of this idea of selective pressure. You would have plasma cells in your bone marrow, and one of them acquired this abnormality that gave it a growth advantage, and then that gave rise to the dominant population of plasma cells. And then in that group of plasma cells, there was one cell that acquired yet another abnormality that gave rise to an even higher risk group or more resistant, more biologically aggressive group of plasma cells. And basically what was happening was that over time, you were gradually developing this more aggressive clone of plasma cells. And so the idea of this, sort of this model is that you're trying to prevent the acquisition of these single higher risk molecular features, and earlier treatment hopefully would suppress the overgrowth of a new, more difficult to treat or aggressive clone. In reality, it's a lot more complicated. Not only is there the acquisition of higher risk molecular features, but it's typical that there isn't just, there might be a dominant clone, but not just one clone. And so what tends to happen over time, and this is really more the case in progression to true myeloma and then myeloma that has become resistant to therapies, is that you develop multiple clones, each of which has high-risk molecular features, and those molecular abnormalities may be different in different clones. And so the problem that we face in myeloma isn't just that something hard to treat has happened in one cell, but different things that are hard to treat have developed in multiple cells. And so you reach the point where there is not a silver bullet anymore. There's not this one target you can go after that's going to take care of all of these different high risk clones. Your chance for that moment, arguably, may be very, very early. It's not just that the cells are more susceptible um, to treatment because of the types of molecular changes, but it's the breadth of molecular changes that have developed. Um, so um, this is actually not a new slide. This slide references some studies in smoldering myeloma that were presented as abstracts at a meeting a couple of years ago, the ASH meeting. And basically, these studies, I think, the reason I pulled this slide out of an old talk was because it illustrates differing philosophy, and in a way, so different philosophies about treating, and very, very different intensities because of those philosophies. So the first one, that's called the Centaurus study, that was reported by Dr. Craig Hoffmeister, who's at Emory University now, but was at Ohio State when the study was done, looked at the idea of just using daratumumab, which is an antibody that targets a protein on on human plasma cells, as a single agent. And they looked at 3 different schedules, and it wasn't given in combination with anything else. And basically what they showed was that in about half of patients, you could induce a response. These responses were almost never curative, not curative. These responses were almost never put patients into a complete remission. So were deduced to not be curative or even potentially curative, but could be hoped might suppress the progression of smoldering myeloma to myeloma. And actually, the basis for that assumption is that that's also sort of exactly the kind of outcomes we saw in the Revlimid studies, the lenalidomide studies that I showed you. Patients generally didn't go into a complete remission, but a sizable percentage of them had responses, and it was therapy that many patients could stay on for extended periods of time. And the idea was that it would suppress progression, and so might be best suited to treat, might be a good strategy for treating smoldering myeloma, because you've acquired at least fewer or a smaller diversity of molecular abnormalities in smoldering myeloma. But because it doesn't eradicate the myeloma, these early clones still may permit those more complicated symptomatic clones to develop later. Now, in contrast, there's the GEM-Cesar study, which was a Spanish study, and this looked at using a 3-drug standard myeloma combination for 6 cycles to treat the smoldering myeloma like symptomatic myeloma, followed by a stem cell transplant like we use for standard myeloma, then 2 more cycles of this 3-drug combination, and then 2 years of Revlimid maintenance, which sounds like a lot of therapy for an asymptomatic disease. But what we saw were very, very different clinical outcomes. So in contrast, when compared to the single agent milder daratumumab study, what we saw was the response rate was basically 100%. Almost all the patients had high quality responses, with the majority achieving complete responses. And even in the first early report of the GEM-Cesar study, we saw that a sizable percentage of patients got to something called MRD negativity, which means not only didn't they have a measurable M protein in their blood anymore, or an excess of plasma cells that we could see with our eyes under a microscope, but they had less than 1 in 100,000 cells. Out of every 100,000 cells in their bone marrow that was analyzed, less than one was a myeloma cell. So this is a very deep response. And then they updated this data after more patients had been enrolled, and ultimately showed that about 2/3 of patients achieved these super deep responses. Okay, these super deep responses. And this was associated with 92% of patients being free of progression to myeloma at 35 months. Okay, which is not what we would expect. Remember, 16% per year for high risk smoldering myeloma, right? And so we would have expected something more along the lines of 40 or 45% at 3 years progressing, maybe even higher. So this is a sizable reduction. And we don't know yet whether or not some of these MRD negative patients may never have reemergence of their clone or not. We don't know that yet. Okay, so in order to even have the potential to have cured a patient, you have to eradicate all measurable evidence of disease. And so this is an example of a curative approach, potentially, although it remains to be seen, how many, if any, of these patients will actually ultimately be cured. I did include one comment here. I made a sort of a passing comment before that our criteria for myeloma has evolved, and it includes looking for evidence of bone disease using more sensitive tests. This was a great example of that. So they originally screened over 100 patients for this study, but 36 patients out of that over 100 who were screened, so somewhere between a quarter and a third of the patients screened actually failed screening, because the imaging testing that was required to get onto this study actually showed unexpectedly that they had skeletal disease. So so finding lytic bone disease depends a little bit on how hard you look for it. And it's very important. This underscores a key management point that I always make with patients, is that if we're going to call you smoldering myeloma, we shouldn't call you that on the basis of negative x-rays. It should be, imaging should include best available skeletal imaging. That should include something like a whole body CT or PET CT or spinal and pelvic MRI testing to look for bone disease that you would miss with less sensitive x-rays. So I'm going to sort of close out by just pointing out some noteworthy trials that are ongoing now or about to be opening. So you've heard about the PROMISE study, which is the screening study that Dr. Gobriel is doing, and she's doing a companion study called the P-CROWD trial, which basically is following patients who are established as having a monoclonal plasma cell disorder like smoldering multiple myeloma, or MGUS, or related conditions like smoldering Waldenstrom's macroglobulinemia, which is a smoldering pre-lymphoma condition that also has an M protein. Those are huge efforts, and those are trials that any potentially eligible patient can enroll on, get involved with just by visiting the websites for those trials, because they're really blood sample driven tests. So those are worth looking for. There are some randomized trials ongoing or planned. Remember, I already showed you that lenalidomide compared to observation improves outcomes in high-risk smoldering myeloma. There's another study comparing daratumumab, the monoclonal antibody that I discussed previously, against observation in the same patient subset. There's another study, which I think might even be a more important study, which is looking at the addition of daratumumab on top of lenalidomide and dexamethasone in high-risk smoldering myeloma. That's particularly attractive because we have randomized trials in symptomatic myeloma, both newly diagnosed and previously treated, that definitely showed when you add daratumumab to lenalidomide and dexamethasone, it improves outcomes. And so you could extrapolate that it might be very helpful in this pre-symptomatic stage, and almost assuredly will increase, will change what I said about patients getting to complete remissions. I think it's highly likely that if you treat patients with lenalidomide, dexamethasone, and daratumumab, that you will start seeing patients get into those super deep responses, because that is what we saw in the treatment of symptomatic myeloma. There's another study looking at iberdomide, which is another drug in a family of drugs called CELMoDs, which is sort of a next generation drug that targets cereblon, which is a target of lenalidomide. Iberdomide is a more potent drug than lenalidomide, or the other drug in that class that's in general use, pomalidomide. And so iberdomide plus or minus dexamethasone in intermediate or high risk smoldering myeloma also is a very attractive trial. It's another oral agent. And then the last two trials that I mentioned here are interesting. They're not randomized, but they are, and they're not as large as these other studies that I mentioned, but they're interesting because they're repurposing drugs that are used for other purposes that are known to have very manageable side effect profiles, to see if they can sort of capitalize on key aspects of our understanding of plasma cell biology. So metformin is a drug that's commonly used to treat diabetes, and leflunomide is a drug that is used to treat rheumatoid arthritis. And these are being studied in these pre-symptomatic myeloma conditions, and will be something that we look forward to seeing the results of. So to conclude, I think this ran a little bit longer than I said it was going to. MGUS and smoldering myeloma have differing risk of developing into myeloma, but even within each group, the risk varies. And it's important to understand that, partly because if we were going to treat any of these patients with therapy outside of the setting of a clinical study, high-risk smoldering myeloma is the one that for which there has been some evidence of benefit, and that would be Revlimid. It's not clear that single-agent Revlimid therapy will ultimately be the optimal approach to these patients. And as I pointed out, there has been more interest in intensive therapy lately, not 100% interest across the board, and certainly not all patients are interested in getting that kind of intensive therapy, but there is interest in using that kind of full blast myeloma therapy on these early patients to try and deeply, deeply suppress the plasma cell clones, and hopefully permanently suppress or even eradicate some of the clones that will ultimately, or subclones that will ultimately give rise to a difficult to treat or incurable myeloma. And as such, I would encourage patients with MGUS or smoldering myeloma to check out whether there are studies potentially available for you, and if participation interests you. And I think that is my talk. So I will stop sharing my screen now.
Great. Thank you, Dr. Zonder. That was so fascinating. I'm just blown away by the results from that. I wrote it down, the Gem Cesar
Yeah, it's
trial.
totally different type of outcome than what we're used to seeing from previous trials.
Yeah.
And it really reflects a totally different treatment philosophy.
Yeah. And I'm amazed, the stem cell transplant going in with just that high risk smoldering, how well the response was after just the stem cell transplant. That's exciting just to see that kind of progress being made in the precursor world. We have lots of great questions. One of them was several of the patients arrived late and aren't aware of what that 2220 means. So
Yeah,
if you could review what that
right.
means.
So
And
2220 has to do with so it refers to three easy to measure markers in a person with smoldering myeloma. The first 20 is the percentage of plasma cells in their bone marrow. The 2 refers to the level of their M protein in grams per deciliter. And the other 20 is the free light chain ratio. And again, free light chains come in sort of two basic types, kappas and lambdas. And so if you have an IgG kappa myeloma, your elevated light chain will be kappa almost always. And if you're IgG lambda or IgA lambda, then your light chain that will be elevated is the lambda. And so when we talk about the ratio, we're comparing the level of the involved free light chain, the one that's related to your myeloma, versus the level of the light chains that aren't. So
Okay.
that's what 2220 is. And if you have 0, 1, or more than 2 or more of those, that's how you sort of plug patients into low, medium, and high risk.
Okay, because that was another follow-up question was, to be high risk, do you have to meet the entire criteria of two?
No, 2 out of 3 is enough.
Okay, great. So there are a couple questions regarding if these treatments are effective for these high-risk patients, why not move it down the line to people who aren't high risk?
Right. So that's the tricky part, right? So if you think about it, right? I mean, I'll just take the bluntest example. Okay. So if you look at the transplant-related mortality risk, okay, from high-dose melphalan, if you look at that at the best centers, the risk of transplant-related mortality, which means fatal complications in the first 100 days after transplant, it's about 1 to 2% at almost all centers, almost all high-quality, high-volume centers. Those are the best centers. Okay. So if you think about that, like, would you take a 1 to 2% risk of fatality related to the therapy if you have low risk MGUS, where your 20-year risk of progression to myeloma is only 6%?
Right.
That right. I mean, and it gets, obviously, as you move up the risk profile in MGUS and then into smoldering myeloma, and then intermediate high risk smoldering, right? Obviously, that the answer to that question, it gets grayer and grayer and grayer, and it's harder to weigh those things out. When your risk of progression to myeloma starts getting quite high, and myeloma, you can't, then you really can't just figure, mortality into the question. I mean, you're talking about symptoms and the need to start treatment indefinitely and things like that. I mean, it's, so it doesn't make as much sense to subject a super low risk patient necessarily to therapy that actually could have permanent or even life-threatening complications to it.
Yeah.
So
Yeah.
I think an average MGUS patient probably wouldn't be willing to risk the peripheral neuropathy that comes with myeloma therapy, the hospitalization rate, the transplant-related mortality. I mean, it's, so it's not for everybody, right?
Yeah, that makes sense. That's a great answer. Thank you. Another question was, I saw this often in our queue, is we talked about how with the CRAB criteria, it can indicate your high risk. What about certain genomic factors or translocations? Does that indicate risk?
Right. So the short answer is yes, probably, but high risk cytogenetic features right now are not generally a primary decider about when to initiate therapy. They that's in a way that's also true to some degree in myeloma, right? We know that certain cytogenetic abnormalities are considered higher risk, and it does in some cases affect the therapy that we use, but for the most part doesn't, right? I mean, for the most part, patients get, a three drug combination regimen, increasingly with an antibody. I mean, with or without an antibody added, but that's generally standard frontline therapy, right?
Mhm.
And it doesn't, we know that high risk cytogenetic features may impact how well or how long that therapy works, but it
Right.
doesn't actually really change what we lead with.
Yeah, I think it's interesting. I think we might see, in the future that we can get more targeted therapies for translocations, but that's definitely not something that we're seeing.
Well, I think
Well,
that that's true. So I think like the 1114 subset as a great example,
yeah,
um, that's a group that is in myeloma particularly sensitive to venetoclax therapy, and that may be a sort of targeted agent that one theoretically might be able to use in that subset of patients
right,
early,
right.
if you have it. I mean, that's an open question. The it is unclear whether introducing targeted agents um, in the early setting will ultimately change outcome the way intensive therapy may change outcome compared to less intensive therapy. We'll see. I mean, a huge interest in this
Yeah,
particular
yeah,
field,
yeah.
in this particular idea. The Multiple Myeloma Research Foundation, the International Myeloma Foundation, a lot of the European working groups. I mean, there's a huge amount of effort in this like precision medicine idea,
Yes. Mhm.
and I'm certain that the PROMISE study will contribute to that understanding. I mean,
Yeah,
it's a great opportunity to learn more about that.
I completely agree. There's a couple people asking about the Ascent trial. I don't know anything about that trial. Do you?
Which trial?
Ascent. It's ASCENT.
I'm not sure. I'm going to plead ignorance. I'm embarrassed to say, but I don't know if that is not the. Um, I'm not sure if that. I'm not sure. I don't
That's fine.
know
That's totally fine.
which study.
Um,
I often find myself behind in terms of the name, like the name of the trial, rather than what
right.
the
What the
what
judge
the
is
trial's
using.
studying.
Mhm.
If anybody on the if any of the viewers on this can just clarify for me
What trial?
clarify for me that trial, I can share my opinion about what that trial is asking or studying.
Okay.
I don't know the name of the Ascent
No
trial. I
problem.
apologize.
There's a couple more questions I know we're going over, but I think there's a lot of good questions going on
Mhm.
here. So, one of the questions was that they've heard about a personalized vaccine study out of MD Anderson. Do you see these approaches as a future direction, these vaccine studies and treatments? Do you see that as a future direction with the possibility for a cure?
So I think cancer vaccines, are just one facet of immunotherapy that is being studied.
Hmm.
I think, um, I mean, I do. I think antibody studies. I think vaccine studies. I think checkpoint inhibitor studies. Um, I think there are numerous immune approaches which have promise, and frankly, um, again, this smaller clone, less um, molecular heterogeneity, within the clones, less subclones, like that probably is actually the right um, time to try and induce an immune
Hmm.
response. It'll be easier to induce an immune response. First, because the plasma cell biology is different, but also your immune system is very different at this early stage than it is after you've been treated for full multiple myeloma for a long time. And
Yeah.
so there is evidence. There are some researchers who have shown evidence, first of all, of sort of immune escape as one of the mechanisms for progressing from smoldering myeloma or MGUS to full-fledged myeloma, but also it's described that patients develop what's called like immune exhaustion over the
Mhm.
course of their illness, and it may make them less able to respond to things like even just antibody therapies. So, yeah,
Yeah.
I think there is a potential promise for those kind of immune approaches in this early space.
Okay, great. Does IgA and IgG share the same risk of progressing in myeloma?
Um, so um, the in the Mayo 2220 criteria, they they tease out IgM as a separate entity, but IgG and IgA, they do not distinguish between those. They don't insist, like they don't, it doesn't only apply to the IgG patients. We know in myeloma that the risk of high risk cytogenetic features may not be the same in IgG and IgA myeloma. And that may account for some of the differences seen, but it's not just the M protein per se. The reason IgM is teased out, though, is because what patients with IgM MGUS are at risk for is really mostly lymphoma and a specific subtype of lymphoma called Waldenstrom's macroglobulinemia. Another name for that is lymphoplasmacytic lymphoma. Um, so it's really it's not, IgM myeloma is extremely uncommon,
Mhm.
and so those patients that really do have a different biology. Um, so IgM is different. IgA and IgG,
Would probably have the same risk.
yeah.
Okay, perfect. We've gotten several comments on the ASCENT trial. Basically, it's combination of Darzalex, Kyprolis, and Revlimid in high-risk smoldering multiple myeloma.
Okay. So Darzalex, Kyprolis, yeah. So that's um, I that that is a similar strategy that's being explored by adding Darzalex to Darzalex is daratumumab,
Right.
and it's the similar idea to adding it to lenalidomide. Um, Dara is now approved in combination with pretty much all of the standard myeloma drugs, and Kyprolis or carfilzomib is one of them. And in that study that I showed you, the Gem Cesar study, it was KRD. K is Kyprolis, which is
Mhm.
carfilzomib,
Mhm.
and that's simply a potent drug in the Velcade or proteasome inhibitor family,
Mhm.
and so I think based on the fact that we know DARA improves outcomes when added to Kyprolis in active relapsed myeloma, and based on the fact that the Kyprolis based Gem Cesar study and others have shown that you can induce really deep responses. There have been other, um, there's been work on the American side also with Kyprolis based therapy.
Mhm.
Ola Langren, even back when he was at the NIH, which is two institutions ago for Ola Langren at this point. And, he was looking at Kyprolis-based therapy back then, right? And so, this is, I think that's fine. I think that's an area of research that's perfectly, ripe for further work.
Yeah.
And it's hard not to be enthused about the idea of adding, CD38 antibody treatment to, um, to whatever backbone you want to try in the smoldering space if you're trying to deepen responses. There's
Yeah.
also a study ongoing or soon to be going, I believe, in Europe using isatuximab,
Oh, interesting.
which is a different CD, that's Sarclisa, that's a different CD38 antibody that's approved in the United States in combination with Pomalyst or pomalidomide for relapsed myeloma. And it was approved based on the fact that it also showed an additional benefit when added to that drug. And
Right.
so that's another example of a CD38 antibody, which hopefully will improve outcomes.
Yeah, I think just to note, this is the exciting part about being in the myeloma field at this time. I mean, as a patient in myeloma, there's so much hope because of the rapid research and progress that's being done in the fields. We talked about immunotherapy earlier. Immunotherapy is so exciting, just the treatments that are being developed, the progress that we're seeing. It's amazing. We have an immunotherapy chapter that meets by month or like every other month, and it's been so exciting to learn more about the treatments and also what's on the horizon of what's to come, because we're just making, well, I'm not making it. You as multiple myeloma specialists are making incredible progress in this field, and it's really exciting. One last question for you. When a patient comes in to you, when do you decide, okay, I'm going to recommend, I'm talking about a high risk multiple myeloma patient, smoldering. Oh my gosh, I can't talk right now. When a high risk smoldering myeloma patient comes to you, when do you recommend a clinical trial and when do you automatically start treatment for that patient?
Well, so every first of all, it's a unique discussion with every patient, and every patient brings their own views and desires about it to the table. And it's the key is honestly making sure that everybody knows the data that they need to know to make a decision, an informed decision, right? It's the current available data. I mean, again, sort of, you know, divorcing myself and ourselves from the idea that you have to get every patient in a complete molecular remission. Okay, that's we assume that will turn out to be a good thing. But if we're just talking about the randomized studies that have been done so far and who benefits from therapy, really we've proved that high-risk smoldering myeloma patients who meet the criteria for those studies benefit.
Mm-hmm.
Now, I also, feel that you can easily make the case for someone who does not yet meet the criteria for high-risk smoldering myeloma, but whose M protein or light chains are increasing in a sustained way, particularly if it's increasing quickly. It's, I also am inclined to treat those patients. I think the harder thing about that, though, right, is, those patients might actually even be worse as a group. By that, I mean their disease might be worse, right? It's you have something that's on the move, already. And even though it doesn't meet those high risk criteria, it's moving, it's changing. It's something is progressing to make the M protein go up. And it may not have reached those arbitrary cutoffs that we use in the 2220 criteria, but its trajectory is going in that way. That may actually, mean something different biologically. I think it actually does, than somebody who meets 2 or 3 of those criteria, but it's been sitting there and has been stable for some established period of time. I saw a patient 3 weeks ago who met the definition of high-risk smoldering myeloma 24 years ago. And, I pretty much what we talk about is how his squash game is going when he comes in to see me every, 2 or 3 times a year, because it's, we monitor his lab work, but he now has a 2-decade plus track record of, disease stability. So clearly his risk is not the same as a newly diagnosed smoldering myeloma patient who you don't have that 24-year movie, you have a snapshot at the beginning of it, the first frame, right? So, I recommend therapy for high risk patients, or at least I talk to them about whether they want therapy. You can justify off-study therapy in that group, and in patients who have this that this smoldering myeloma in transition to
Mhm.
something, to, that's worsening. And then patients with lower risk disease, honestly, I mean, outside of a study, I'm not really an advocate of starting therapy. Um, a common question is things like supplements, and we talk about the things that there's data for, maybe data for like turmeric or green tea extract, but you know, I don't come out strong for
Right.
those. I just tell them about the data. You know.
Okay. And then just a sub question, how often do you recommend that MGUS and smoldering patients get tested and come in and get their work done?
So a a low risk MGUS patient is probably somebody who can be followed by their internist and may not even need to follow up with a hematologist. A intermediate or standard risk MGUS patient, I generally offer them the option of following up with me annually,
Okay.
or with their internist, and call me if you need me. Um, for I follow smoldering patients. I don't generally turn smoldering patients back to just their internist to follow up. So for them, every depending on whether they're high risk or not, every 3 to 6 months is how often I follow them. In the first year, pretty much any of them I will follow every 3 to 4 months.
Okay.
And then after that first year, depending on how things are going, we may space it out a little bit.
Okay. Awesome. Well, thank you so much, Dr. Zonder. That was a great presentation, and I feel like you moderated or answered those questions really well. So thank you. We're going to finish up today with just a couple outro announcements, and then we'll be done. So our next event will be on September 2nd. We'll be hearing from Dr. Jens Hillengas. He is going to be talking about the fitness study that he's doing for precursor myeloma patients. And one of the questions that he's researching is, how does exercise and overall fitness affect myeloma progression? So again, that's September 2nd, 7 p.m. Eastern. You may be interested in any of these upcoming events within the Myeloma Crowd Community Program. Because we're over on time, I won't go through them, but we will be sending out my slides as well as the recording in our follow-up email within 48 hours. I will also include different abbreviations that Dr. Zonder used. I saw some questions in the chat about that. And then I'll also try to include as much information as I can about the trials that Dr. Zonder mentioned, as well as how to find these trials using clinicaltrials.gov or HealthTree Cure Hub. Thanks again to our sponsors, Bristol Myers Squibb, Amgen Oncology, Karyopharm Therapeutics, AbbVie, Adaptive Biotechnologies, Genentech, Oncopeptides, Takeda Oncology, and thank you for joining us today, for helping us build a strong myeloma crowd community. I appreciate you and hope you have a great rest of your day. Thank you so much.